For those who have gotten coronavirus and have recovered and developed some degree of immunity, dendritic cells have probably played a role. As the major antigen presenting cells of the lungs, they were probably among the early cells to encounter coronavirus. They probably produced local hormones known as interferons, which boosted antiviral cellular responses. They probably activated cytotoxic T lymphocytes by using their MHC1 proteins, which had attached peptides from, say, the viral spike proteins. They probably activated helper T lymphocytes with their MHC2 proteins in a similar fashion. They probably left the lungs and traveled to lymph nodes, where they helped to train lymphocytes, which would then boost the adaptive immune response, including the humoral response, where antibodies against coronavirus were produced. Presumably, dendritic cells will play an important role in the immunity which results after a vaccine for coronavirus. Now, given that the first two vaccines which were approved involve viral RNA wrapped in lipid nanoparticles, it is possible that dendritic cells simply take up the foreign RNA because the lipid nanoparticle fuses with the lipids of the cell membrane, and that this is similar to what could occur in many cell types, perhaps most cell types, and the dendritic cell might not be that special in this regard. The expression of foreign RNA inside a cell could be seen as an accident. The vaccine's lipid nanoparticles accidentally get introduced into the cell because the lipids of the nanoparticles fuse with the lipids of the cell membrane. But it is known from mouse studies, as RNA vaccine technology was originally experimented with, that actually naked RNA can also produce an immune response. This RNA would not have these lipid nanoparticles. This raises the question, why would cells of the body take up foreign RNA, which could come from an infectious agent, and express it? Isn't this almost inviting infection? Actually, it is a fairly recent discovery that dendritic cells actually take up foreign RNA and express it as part of normal immune responses. Imagine, for example, that there is an infection and that a number of cells die in an infected tissue. Dendritic cells come to the area and they perform phagocytosis, perhaps of infected cells, but certainly of debris and proteins which may originate in dead cells or the infectious agent. They could also then uptake foreign mRNA. By expressing the mRNA and thus producing proteins from an infectious agent in the dendritic cell, which then could be displayed on MHC proteins, the dendritic cell would then be able to alert other immune cells to the foreign proteins, so that they would then mount an immune response against them. And this would include proteins from pathogens that the dendritic cell did not encounter. Consider, for example, that the majority of viruses would not infect dendritic cells, but these dendritic cells could nevertheless express viral proteins to help the immune system fight these viruses, even those that did not infect the dendritic cell. There is no doubt that dendritic cells play a significant role in our immunity to coronavirus. However, their unique ability to take up foreign RNA and debris with the idea of expressing this to immune cells and traveling to where the immune cells are maturing, this could be significant in the success of coronavirus RNA vaccines promoting immunity to coronavirus.